So we're going to continue talking about neurophysiology today, but let's review just a little bit. Yesterday we ended on talking about resting potentials. So remember when we were talking about resting potentials, we said that it's due to ion channels and it's due to sodium-potassium pump. And so what's going to happen is we're going to be constantly moving sodium and potassium into and out of the cell. And so this sodium potassium pump, remember, it pumps 3 sodiums out, and you need to remember this, really important, 3 sodiums out and 2 potassiums back in. And so remember, that's a 3-to-2 ratio. And because they're both positively charged, that means it's 3 positives on the outside compared to only 2 positives on the inside. And remember, what's going to happen is we're going to set up a sort of a steady state where the pump pumps these ions, and then we're going to create gradients, and then sodium is going to leak down its gradient. It's going to come in through the leak channels, and potassium is going to leak down its gradient. It's going to go out through the leak channels. What we're going to get is this steady state. And if we measure the steady state, remember it's measured in volts, which is a measurement of electrical potential, we get across the membrane a voltage of about minus 70 millivolts. And so if we look at that, this is what it looks like with those ions. And so here's our sodium potassium pump right here. And remember, again, 3 sodiums out, and then turns right around and pushes 2 potassiums in. And so we're going to build up sodium here on the outside, and we're going to build up potassium on the inside. But what's going to happen is when you create those gradients, since we have these open channels for both sodium and potassium, sodium's going to leak down its gradient, so sodium's going to tend to come back in. And potassium's going to leak down its gradient, so potassium's going to tend to go back out. But as soon as that happens, remember, the pump is going to turn around and again pump 3 sodiums out, and it's again going to bring 2 potassiums back in. But as soon as that happens, we're going to get leaking. Sodium's going to leak in, potassium's going to leak out. But as soon as that happens, the pump is going to move them in the other direction. And so what's going to happen is we have these 2 opposing forces. And so we're going to set up this sort of steady state where there is more sodium on the outside, more potassium on the inside. The outside is more positive and the inside is more negative. So the 2 sides of this membrane are very different from each other, so it's called polarized. And we can measure the difference in voltage. We put a voltmeter, and we take one electrode and we put it on the inside next to the membrane. We take another electrode and put it on the outside next to the membrane, and we get this voltage of about minus 70 millivolts. Again, that is a steady state, which means it's not going to change, and we call it resting potential. So if we measure resting potential, it looks like this. If we graph resting potential, it looks like this. So here's our voltmeter right here, and there's our 2 electrodes. Again, one on the inside, one on the outside, and here's that voltage, minus 70 millivolts. And so if we graph it, because it's a steady state, it's not going to change. It's going to stay at minus 70. And that's exactly what we see here. So this is resting potential. And that's about where we stopped last time. Does anybody have any questions about resting potential? Again, here's that sort of summary slide that kind of shows everything that's going on in the same slide. You can see the pump, there's 3 sodiums, 2 potassiums, there's the leak channels, There's the voltage. It's using ATP because it's active transport. All of that is in this one slide. So it's a nice place to come and look at what happens during the steady state. And then if you look at the actual numbers for sodium and potassium, remember they're not going to change very much. Sodium is much higher on the outside, like 150. Much lower on the inside, only 15. That's because this pump is pumping it out. It also, though, remember, brings potassium in. So potassium is much higher on the inside and much lower on the outside. And so you can see that there's a gradient here. And again, that gradient is going to cause them to move through the leak channels. And remember, it always only goes from high to low. So sodium's going to go this way from high to low. Potassium's going to go this way from high to low. And so we're going to get this diffusion through the membrane, through those channels. And so there are these two forces that are opposing each other. And these two forces are what's going to create and maintain resting potential. So one is the sodium-potassium pump, and the other one is the leak channels, the leaking of the ions through the channels. That's what's going to create and maintain resting potential. And so if we graph resting potential, remember it's a straight line. That's resting potential. And resting potential is everywhere. It only shows it right here, but there's resting potential here and here and here and here. Resting potential is everywhere. But resting potential, remember, is just this straight line. And if it never changes, you cannot use resting potential for a signal because it just doesn't change. So to do signaling, we're going to have different kinds of potentials which do change this line. And so these signaling potentials can be used for lots of things. They can be used to send information, receive information, integrate information. And what's going to happen here is we're going to change the ion movements. So we're going to make more ions move than what moves during resting potential. And when you alter the concentration of the ions, we're going to see a change in the graph. So look at this. Here's resting potential right here, minus 70. And so look, it's like a line. Nothing's changing. You ever watched a movie where, I don't know, it's sort of a spy movie and there's a boat out here and over here's the shore over here, and you see all these people standing here and they're waiting for a signal from this boat. Well, it's pitch black outside. They can't see anything. But then finally some guy on the boat takes a tiny little flashlight and just blinks it 1, maybe 2, maybe 3 times. Well, that light, because it's pitch black out here, that light is very, very visible to the person on the shore. And that's exactly what these signaling proteins are like. So look, this is like pitch black. There's no change. But all of a sudden, here's this little tiny burst of light. In other words, if we change this voltage, what we're going to see is this little tiny change in voltage, and it's like a signal. And then here's resting potential again, and it changes again, and again, it's like a signal. Does this make sense to everyone? So what we're going to do is we're going to have to change this voltage to produce this signal. And so we can't use leak channels because leak channels are always open. So what we're going to have to use are gated channels. Remember, gated channels can be open or closed. And if they're open, it lets ions move very quickly across the membrane. And when the ions move, remember, they have a charge on them. Sodium has a +1, potassium has a +1, they have a charge. And so when those ions move, the charges move with them. And we create this little electrical current. There are two kinds of these signaling potentials, two kinds. One kind is called graded potentials, and the other one is called action potentials. Graded potentials, action potentials. Well, graded potentials don't travel very far, and so they're for short distance communication. They don't travel very far. But action potentials travel really, really far, and so they're good for long distance communication. So graded potential, short distance, action potentials, long distance. Well, remember we have this resting potential, which is at -70 millivolts. Well, in order to have these graded potentials, this has got to change. Remember, this is like pitch black dark. It's got to change. And so something is going to have to make it change. And whatever makes it change is called a stimulus. So look, here's our line right here. Well, let me ask you a question. If this line changes, which direction can it go? Can it go up or can it go down? Which one? What do you think? Anybody want to guess? If this line changes, which way can it go? Can it go up or can it go down? Johannan says up. Anybody else want to guess? Well, the answer is it can go both ways. If this line changes, it can change this way, but it could also change this way. We can see that. Well, whatever makes it change is called a stimulus. And so if it makes it change in two different directions, that means that there's two different types of stimuli. If it goes toward zero, here's zero here. If it goes toward or above zero, it's called an excitatory stimulus. If it goes in the other direction, look, it's moving further away from zero, it's called an inhibitory stimulus. So we have two different kinds of stimuli that are going to make this graph move. So let's look at these two types of stimuli. Let's look at an excitatory stimulus first. So what happens with an excitatory stimulus is the graph is going to move in this direction. Well, remember what our membrane looks like. So here's our membrane, and here's our pump. Here's another pump. Remember, it pumps 3 sodiums out, and it brings 2 potassiums back in. And it does that over and over and over again. Same thing over here, 3 sodiums out and bringing 2 potassiums back in. But then remember, we also have these leak channels. So we have sodium leak channels. So here's sodium leak channels. And then we also have potassium leak channels. And here's potassium leak channels. And so what's going to happen is when sodium builds up on the outside, sodium's going to tend to leak back in. It's going to come back in through these channels. We already know all this. It's going to come back in. And potassium's going to tend to go back out. And we're going to set up this steady state. And that steady state is this. It's resting potential. So it stands to reason then, if something makes this line move, it has to be these ions, because that's what's creating resting potential. And so if we look at excitatory stimulus, it's one of these 2 ions. And the ion it is sodium. So look what happens. If we open the channel for sodium, this is a gated channel now, not a leak channel. Remember, there's more sodium out here on the outside. There's more potassium on the inside. If but we're going to open a gated channel for sodium. And so as soon as you open that door, that means that more sodium can enter. So sodium is going to come in faster than it usually does. Well, what kind of charge does sodium have on it? What kind of charge is that? It's a positive charge. So look, here's my resting potential. I'm at minus 70 millivolts. But I'm adding positives because positives are going to come in with this sodium. So what's going to happen to my graph here? It's at minus 70, but if you add positive numbers to that, which direction is it going to go? Is it going to go up or is it going to go down? It's going to go up. That's exactly right. And if it goes up, remember, it's called an excitatory stimulus. So that's exactly what this looks like. Sodium comes in faster than it usually does. So right here is where that stimulus opened those gated channels. Right there, we opened the gated channel. And sodium came pouring in. Sodium goes rushing in. And when sodium goes rushing in, our graph is going to go this way. It's going to go up towards zero. And because it's towards zero, it's an excitatory stimulus. Well, remember when we talked about this membrane, we said that if it's at minus 70, we already know this, that the two sides of the membrane don't look alike. The outside has more sodium, the inside has more potassium, the outside is positive, and the inside is negative. And remember we said if it looks like that, you could call that a polarized membrane. Polarized, different. But look, when I move towards zero, if I move in this direction, I'm moving towards zero. At zero, the two sides are exactly the same. They're exactly the same. In other words, if you were to draw it, you have just as much sodium inside as you do outside. Just as much potassium as you do outside as you do inside. And the charge on the two sides is exactly the same. In other words, there's no voltage. It's not polarized anymore. And if it's not polarized, no voltage, well, if I move toward it, I'm going from polarized to not polarized. It's a depolarization. I'm removing the polarization. So an excitatory stimulus moves it towards zero. And so that's called a depolarization. So if we look at this, here's our stimulus right here. That's an excitatory stimulus. And that excitatory stimulus, remember, is going to open gated sodium channels. So sodium's going to go in. When sodium goes in, my graph's going to move in this direction. And if I move in this direction, that's called a depolarization. But look at this. Here's where those channels open. What do you think happened right here? What do you think happened right there? Look, this blue is different. So here's where the stimulus started. This is where the channels open. But look, something happened and I removed the stimulus, and Johanan says that's where they close, and that's exactly right. The channel's closed. But also look at this. I don't stay up here. I don't stay there. This goes right back to resting potential. And I go right back to that steady state. So what is bringing this back to resting potential? What's causing this to happen? Do you remember things that create and maintain resting potential? What are they? Two things. Create and maintain resting potential. One is the sodium-potassium pump, and the other one is the leak channels. So when I close those gated channels, the sodium-potassium pump is going to start pumping the sodium back out. And as soon as I pump the sodium back out, look what happens to my graph. It goes back to where it was. And so I go right back to resting potential. So one more time. Remember, if you look at this, here's our stimulus right here. And that stimulus is going to open gated sodium channels. Sodium's going to rush in. It's going to bring a positive ion with it, or positive charge with it. And I'm going to move towards zero. Again, that's an excitatory stimulus, and it's called a depolarization. And we can see that exact thing right here. So, sodium is associated with depolarization. But look at this. What happens if we do an inhibitory stimulus? Well, an inhibitory stimulus is going to open a potassium channel. So here's my graph again. And remember, potassium's on the inside. So if I open a gated potassium channel, potassium's going to rush out. So look, potassium also has a positive charge on it. And so if I take positive charges out, I'm going to make the inside more negative. It's going to go this way. And that's exactly what this graph does. It goes this way. Well, this is more polarized than it used to be. This is polarized. This is more polarized. And the word for more is hyper. So this is called a hyperpolarization. But again, look what happens. I don't stay here. So what happened right there? Do you remember what happened on the other one? What happened? Here's where those gated sodium or potassium channels open. So what happens right here? They close. And again, how am I going to get back to resting potential? How am I going to get back up here again? What's got to happen? Remember, there's two things that create and maintain resting potential. What are they? Those two things. Sodium potassium pump. What's the other one? The leak channels. And so they're going to, so the potassium is going to start getting pumped back in here again. And as I pump the potassium back in, I'm going to go back to resting potential and maintain resting potential. So if I look at those two things, I get either a depolarization or I get a hyperpolarization. Which ion is associated with an excitatory stimulus and a depolarization? Sodium, that's right. Which ion is associated with an inhibitory stimulus and a hyperpolarization? Potassium, exactly. So if I look at those two things at the same time, you can see exactly what happens. There's my excitatory stimulus. There's my inhibitory stimulus. Remember, this is a depolarization. This is a hyperpolarization. And right here is where the channels open, the gated channels, they open right there, but it's a different channel. Here it's a gated sodium channel. Here it's a gated potassium channel. And then just like we said before, When I close the channels right here, that's what happens. I'm going to close the channels right there. I'm going to use the sodium-potassium pump and the leak channels to get back to resting potential. So Johannes says on the graph, looks like both happens at the same time. These are two different graphs. Is that what you're asking? These are two different graphs. So these are two separate events. You could split it right here. They just put both of them on the same page. Anybody else have a question? Does everybody get this? So, one more time. Excitatory stimulus, depolarization. Which ions involved? Sodium. And what does sodium do? Does it enter the cell or leave the cell? It enters. And it's going to happen through gated sodium channels. It's going to happen right there. We're going to open a gated sodium channel. And then an inhibitory stimulus, a hyperpolarization. Which ion is involved? Potassium. And what does potassium do? Does it enter the cell or leave the cell? It leaves the cell. And again, the way it does that is I'm going to open a gated potassium channel. Does it make sense? Can everybody interpret this graph? Or these graphs? So that's graded potentials. Well, graded potentials are called that because of some things they have, some characteristics. One of the characteristics of graded potentials is that they come in different sizes. So look at this. If I have this whole thing right here, and here is a gated channel, and here is a gated channel, and here is a gated channel, and so on. Well, what happens if I open just this one gated channel? How much sodium do you think can move, or how much potassium could move? Sodium only has one more door to come through than it did before. So would it be a little bit of sodium coming in or a lot? It would just be a little. So if I look at my graph and it moves, is it going to move up a little or is it going to move up a lot? It's only going to move up a little. That's exactly right. But what if instead of opening one channel, I'm going to open two channels? So now there's two ways for sodium to get in. So sodium's going to come in this way and this way. So look, now at my graph, I look at it. Is it going to go up a little, a medium size, or a lot? Is it going to be like it was here? Is it going to be a little bit more? Or is it going to be a whole lot? It's going to be medium. That's right. Well, what if I open all of these channels at the same time? I'm going to open all of them, and sodium's going to come rushing in all of these. And so if you look at that, what's going to happen is the graph is going to go up very, very, very fast. It's going to go up very high. So when I look at these different potentials, if I have a different size stimulus, I'm going to open more channels or fewer channels, and more sodium's going to come in or less sodium. So they vary in size. And the size is dependent on how big a stimulus it is, the stimulus strength. Another thing about these is we already know we can be depolarizing or we can be hyperpolarizing. Remember, an excitatory stimulus makes it move this way, an inhibitory stimulus makes it move this way. So it can be depolarizing or it can be hyperpolarizing. It just depends on which kind of channel opens. Which kind of channel opens if it's depolarizing? Which ion? Sodium, gated sodium. Which kind opens if it's hyperpolarizing? Gated potassium. So it can be either one. It just depends on which kind of channel it opens. And then another thing about this is they don't travel very far. So look, if this is my membrane, this whole thing, and right here's a bunch of channels. So there's a channel, here's a channel, here's a channel, here's a channel. But I'm only going to open this one. So look, let's say it's a sodium channel. Sodium's going to come in. And sodium's going to begin to diffuse in all directions. Because remember, it diffuses from high to low. So I'm going to have a lot of sodium right here, a lot of sodium. But the further I get away, the less and less there's going to be. And if I go very far, there's not going to be any at all. So look, it only traveled this far. That's as far as it got right there. That's as far as it got. So they don't travel very far. They get smaller. They dissipate the further you get away from the stimulus, which is right here. They also don't last very long. As soon as this channel closes, the pump is going to pump all that back out. And it's going to disappear. All that sodium is going to go away. And pretty soon, there's not going to be any extra sodium in here at all. So they don't live very long. They're not last very long. And then the last thing is the type of channel that's involved. Remember, they're gated channels, but they're chemical gated or mechanical gated, not voltage gated. They're chemical or mechanical, but not voltage gated channels. So those are characteristics of graded potentials. So look at this. These are all graded potentials. So look, they vary in size. So look, they vary in size. And I bet if you look at this, you can figure out the size of the stimulus. Look, that's how much change there was. It was a little stimulus. But look, that's how much change there was. It was a medium-sized stimulus. That's another medium-sized stimulus. But look, it lasted longer because the channel stayed open longer. But look at this one. That is a large stimulus. Right there's a large stimulus. Same thing on this side. Small stimulus, medium-sized stimulus, large stimulus. And if you look at this, look, they can be depolarizing or they can be hyperpolarizing. Depolarizing, remember, goes towards zero. It goes this way. And if this one goes away from zero, it goes this way. And I hope by now you can tell me which ion was involved with which. Which ion is involved here? Which ion? Sodium. Which ion is involved here with these? Potassium. Any questions? Remember, they also don't travel very far. They dissipate. So right here is where the stimulus is. That's where the stimulus is. So right here is where sodium is going to come in or potassium is going to leave. That's where it happens. But again, it's going to spread. But the further you get away from here, the less and less there's going to be. And they use color here to represent that. So right here is very red. And then there's this lighter red. And then finally it goes orange. And then finally over here it's yellow. They're using it to show you that there's less sodium coming in. And if there's less sodium, the change in this potential is going to be less. Over here it changed this much, whatever that is. But here it didn't change at all. It doesn't travel very far. They're short distance signals. So look, here's a stimulus. I don't know what this is, but it might be something like a pen or a needle. So look, it pokes somebody. And there are channels right here that are going to open. Which kind of channels do you think this is? Do you think it's chemical gated channels that open, or is it mechanical gated channels that open? Which one is it? It's movement, it's pressure. So is it chemical or mechanical? It's mechanical. In other words, mechanical gated sodium channels open, and sodium's going to come in. And then it's going to spread. But look, the further you get away from that point where the pen was, the less and less sodium there's going to be until there's no extra sodium over here, no extra sodium. So this is the distance that this traveled. It traveled just to here, and that's it. It gets smaller the further you go. It dissipates. Any questions about that? So remember, if we get chemical gated or mechanical gated, we're going to get these graded potentials. So let's review chemical gated and mechanical gated channels a little bit. So here's a chemical gated. Remember, there is a receptor here. And there's a chemical, a neurotransmitter that's going to bind. And as soon as it binds, the channel opens and ions can flow. If it's a mechanical gated one, remember it requires pressure. Something has to push on it. But when you do push on it, when there is movement, the channel is going to open and ions can flow. So again, here are these characteristics. They vary in size. Remember, they can be a little one, they can be a bigger one, they can be a bigger one, they can be a bigger one. They vary in size, and the size is dependent on the stimulus strength. So if we look at these, you can see this is a small stimulus. But this one is a big stimulus. The size is dependent on the stimulus strength. We already know they can be depolarizing or hyperpolarizing. Remember, they can go this way, but they can also go this way. They don't travel very far. They dissipate with distance. They don't last very long. And remember, the channels that are involved are chemical gated or mechanical gated channels. These, not voltage gated channels. Just these two types here. So if you look at the graph of a graded potential, it looks like this. So look, there's resting potential, and right here's where the stimulus was applied. Look, chemical stimulus. So what kind of channel opened here? Was it chemical or mechanical? Chemical. And what ion is going to go through it? Sodium. So it's a chemical gated sodium channel. That's what opened right here. And it goes up. And then look, here is where the chemical stimulus was removed. And so what's going to happen is the channel's going to close. And then we're going to use sodium potassium pump and the leak channels to restore resting potential. Same thing over here on this side. Chemical stimulus, but this time a different channel opened. It's a chemical gated potassium channel that opened. Potassium is going to leave. Right here's where the stimulus was removed. And we're going to use this. We're going to do the sodium potassium pump and the leak channels to get back to resting potential. And so if you look at this, when it goes up this way, remember that's called the depolarization. So when it comes back down, it's called a repolarization. When it goes this way, it's called a hyperpolarization. And you would think that this would be called a repolarization. But we already used that. We used it over here. So over this one is called a return to resting potential. So when you graph a graded potential, it can be either one of these. So here, excitatory. Here, inhibitory. Here the ion is sodium. Here the ion is potassium. It's chemical gated. We know that because we're going to use a chemical stimulus. And then we get a depolarization, a repolarization, hyperpolarization, and a return to resting potential. Any questions? So that's graded potentials. And graded potentials then, remember, they move either up or down and so on. Well, if it's excitatory, if it's an excitatory stimulus, it's going to move this way. Well, here's our resting potential. There's actually another potential up here, which is called threshold potential. And threshold potential is where voltage-gated channels become involved. So remember, we're already using chemical or mechanical, but at threshold, voltage-gated channels become involved. And what happens is if you reach threshold, we're not going to get a graded stimulus anymore. It's going to turn into an action potential. And so if you look at an action potential, this is what an action potential looks like. So look at this. Here are a bunch of graded potentials right here. They're graded. Look, they're in different sizes. We got a small right here. We got a medium right here. But neither one of those, small nor medium, got all the way up to threshold. And so the sodium potassium pump and the leak channels just bring it right back down to resting potential. But this large graded potential made it to threshold right there. And at threshold, the graded potential is going to end and an action potential is going to begin. And action potentials look like this. They zoom way up. They go up to about plus 30 and then they come back down. And they come back down even further than before. They all look like this. So if you look at it, once you reach threshold, you're going to get an action potential. So this particular one right here reached threshold. Remember what's going to happen at threshold. The voltage-gated channels are going to become involved. And voltage-gated channels give you not graded potentials, they give you an action potential. And what's going to happen is that action potential, once it's generated, is going to travel all the way down the membrane. Any questions? So we're going to look at action potentials a little bit. I doubt we'll finish, but we're going to talk about them for a while. But let's look at action potentials. So remember we saw this thing, and I used the analogy of a boat. And so here is that pitch black night right here, and then here's when the flashlight went off. And the flashlight, then we can use that as a signal. So this is a signaling protein. And so look at these. Remember we talked about graded potentials and how they can come in different sizes. Those are all graded potentials. And remember they happen when we open a mechanical or chemical gated channel. But none of these graded potentials reached threshold. And so remember, this is where voltage gated channels become involved. But none of these reached the place where voltage gated channels are going to become involved. And so they go right back down to the threshold. And then we stimulate it again, and it goes up a little higher, but it didn't make it to threshold. It goes back down to resting. No matter how close you get, to threshold, if you don't actually get there, it's just going to come back down. But if you do reach threshold, these voltage-gated channels are going to become involved, and we're going to get an action potential. And the action potential is going to go through this whole set of steps. And when you get an action potential, it's going to look like this. But again, you must reach threshold. These did not reach threshold. And so no action potential is going to happen. They're just going to come back down to resting potential, come back down to resting potential. But if you do reach threshold, you get an action potential. And here's an action potential. Any questions about it? So let's look at what an action potential is. So an action potential is a signal we already know. And this is one of those signals that can travel long distances. And this signal appears wherever we reach threshold. Because what happens at threshold is the voltage-gated channels are going to become involved. Threshold potential is where the voltage-gated sodium channels open. And so when you open voltage-gated channels, right there, sodium is going to rush in. And because we're using voltage, all of the channels in this area where the voltage is going to open. And so sodium rushes in like crazy. It's going to go up to plus 30. But plus 30, what's going to happen is those channels are going to close. So the voltage-gated sodium channels open here, and they close here. So during this time when they're open, sodium is going to rush in. But then when we get up here to plus 30, they're going to close. But at the same time they close, the voltage-gated potassium channels open. And as soon as that happens, potassium is going to go rushing out. And it doesn't have close until you get down here to about minus 90. And that's where they close. And then look where we are. We're down here. We have to get back to resting potential. And again, it's going to be the sodium-potassium pump and the leak channels that are going to restore resting potential. Well, every single action potential looks just like this. They all do. Here's another picture. Looks just like this. And so again, different things are happening at each one of these stages. So right here is where our excitatory stimulus is. And it's either going to open a mechanical gated or a chemical gated channel, and sodium's going to come in. That is a graded potential. And remember, if it doesn't reach threshold, it's just going to come back down, come back down, come back down. But if it does reach threshold, right here is where those voltage-gated sodium channels are going to open. Voltage-gated sodium channels are going to open. And sodium is going to rush in. And the channels are going to stay open until the voltage reaches plus 30. And then those voltage-gated sodium channels are going to close, and the voltage-gated potassium channels are going to open. As soon as they open, potassium is going to rush out. Our graph is going to go down. It's going to go all the way down to about minus 90. And that's where those voltage-gated potassium channels are going to close. And then we're going to use our sodium-potassium pump and the leak channels to get back to resting potential. So this is sort of like a practice quiz slide. So you should be able to figure out what all of these things are. One, I don't know who asked that. Johan, this is a graded potential. And remember, graded potentials are going to generate action potentials. But the only way they can generate it is if they reach threshold potential. And threshold potential, again, is when the voltage-gated sodium channels open. If they don't reach threshold, if they're just this big, or this big, or this big, or this big, no matter how big they are, as long as they don't reach threshold, it's just going to come right back down to resting potential. But the second they reach threshold and the voltage-gated sodium channels open, that's when the action potential starts. So these are not action potentials. These are graded potentials. So I'd like you to be able to tell me what happens at number 1, what happens at number 2, all the way to number 8. So you should sit down and write out this set of steps from 1 through 8. And even if the numbers aren't on there, you should be able to figure it out. Right here is the stimulus. Right here, that's our graded potential. Right there is where the voltage-gated sodium channels open. Here is where sodium is rushing in. Here's where they close. Here's where the potassium channels open. Here's where potassium is rushing out. Here's where they close. And right there is our return to resting potential, to get back up to resting potential. And again, that's the pump and the channels. Does it make sense to everyone? So you might see this on a quiz, or you might see it on a test, asking you what happens at 4, what happens at 8, and so on. So here's what those channels look like. So right here is where threshold potential is, because what happened at threshold, which is about -55, is this. That's our voltage-gated sodium channel. Voltage-gated sodium channel. Look, it's open. This one right now is still closed, but we're at -55. So here's our graph right here. Right there is where this happens. And then remember, it's going to shoot up. And it's going to go up to about +30. And at +30, this is going to happen. The voltage-gated sodium channel is going to close, and the voltage-gated potassium channel is going to open. So now what's going to happen is we've gone all the way to the top. When the potassium leaves, it's going to come back down. And it's going to go all the way down to about -90, and then this one's going to close. So if you want all that in words, that's what this is here. So we're going to get a depolarizing or excitatory stimulus. We're going to open voltage-gated sodium channels. Sodium's going to rush in. That's this part here. When we reach +30, what's going to happen is the channels of sodium are going to close, and the potassium channels are going to open, and potassium is going to leave the cell. So that's this phase here. And then when we reach down about -90, potassium channels are going to close, and we're going to get a return to resting potential. And remember, it's the pumps and the leak channels that are going to return us to resting potential. So here's another little quiz-like thing. A, B, C, D, E. So which one of these is depolarization? Is it A, B, C, D, or E? Depolarization. It's B. Which one of these is hyperpolarization? It's D. Which one of these is repolarization? That's C. Which one of these is a return to resting potential? That's E. And then A is just resting potential. And that's what an action potential looks like. It's divided up into these different phases. Again, what happens at each one of these numbers? So what happens at 2? What happens at 2? 2 is threshold potential, but what happens at threshold potential? Voltage gated sodium channels open. What happens at 5? voltage gated potassium channels open. And if you do this multiple times, it just gets easier and easier and easier to do. But you need to practice it. So you need to use this graph, something like it, and practice. Because you will see this on quizzes and on exams. Now, if you're using the PowerPoint, this is a little video you can watch. It's on YouTube. So if you click on that, it'll take you to a little YouTube video about the action potential. So we saw characteristics of graded potentials. Remember, they vary in size. They don't travel very far. They can be hyperpolarizing or depolarizing. We already know all that. Action potentials also have some characteristics. One of their characteristics is that they're what is said to be all or none. All that means is you either get one or you don't get one. It either happens or it doesn't happen. There are no half action potentials. When you get them, they're all the same size. Every one of them goes up to plus 30, and that's it. That's exactly what they do. So when you draw them, they're always exactly the same. Another thing is they don't dissipate. They don't get smaller. They're all to plus 30. So if you look at one, if this is a whole membrane and you look at this one right here and it goes to plus 30, or let's say plus 29 and a half. If that one goes to plus 29 and a half, all the way over here, it's going to be plus 29 and a half. When it gets from one end to the other, it's not going to change at all. They happen when you reach threshold. Remember, if you don't reach threshold, you don't get one. They're all or none. If we reach threshold, we're going to get one. And then they're all the same size. If you reach threshold, the stimulus strength after that doesn't matter because the size is independent of the stimulus strength. It just has to be strong enough to reach threshold. Now, if you don't reach threshold, you're not going to get one because they're all or none. And then there's what's called a refractory period. And the refractory period says that there's sort of a reset period, a reset time. Because think about it, here's what it looks like. Over here, sodium comes in, and over here, potassium goes out. That's not the way our membrane's supposed to be. It takes a little bit of time in order to get the sodium back where it's supposed to be and get the potassium back where it's supposed to be before I can get another action potential. And so there's this period called the refractory period. It's like a reset time. They're a little bit like dominoes. So look, if I knock over dominoes, this one domino has already fallen. And I cannot make it fall again until I reset it back up again. And that takes a little bit of time. There's this reset period. Again, it's called the refractory period. So this domino is refractory, and this one is refractory, and this one is refractory. These are not refractory. They're already set up. So I can get an action potential here, but I cannot get an action potential over here until they're reset. And then one last thing about action potentials is they get recreated over and over down the membrane. So if here's a membrane and you get an action potential, it doesn't go all the way. What happens is this action potential creates another action potential. And that action potential creates another one. And then another one is created, and another one, and another one, and another one. Now remember, they're all the same size. I'm not drawing them that way, but they are. But they get recreated over and over. So look, this one is the stimulus for the next one, and so on. And if it's almost like dominoes again. So look, these dominoes are all set up. They're all ready and waiting. But this one fell over. And when that one fell, it knocked this one over. In other words, it was the stimulus to make this one fall. And then that one knocked this one over. It was the stimulus to make the next one fall. And then that one's going to knock that one over, and so on. So what's happening is all these dominoes are individual dominoes. They're not even touching each other. Look, they're just set up like this. But if I bump this one, if I stimulate this one, they're all going to fall down. And that's exactly the way action potentials are. And then, like those dominoes, there's this reset period, this refractory period. It's a time to reset the channels. So here is the refractory period. So it starts right here when the sodium channel's open, and it ends over here when I get back to resting potential. So that's when this voltage-gated sodium channel's open. And here's when I get back to resting potential. That's this reset period. Now it's divided up into two parts. One part is called the absolute refractory period, and the other one is called the relative refractory period. And then during the relative refractory period, if you have a really, really strong stimulus, the channels will go ahead and open, and you can get an action potential. So, absolute, relative. No way to get an action potential here. No way at all. But here, if you have a big, big stimulus, then yes, you can get an action potential. And then we're back to hit this again. So what happens in 1? What happens at 2? What happens at 3? You should practice this until you don't have to have these numbers, until you can look at it and just figure out what's happening as you move through this graph. And then finally, it's just like dominoes. So look, it's all set up. It's like dominoes are all set up. And they're just waiting. Remember, it's at rest, resting potential. And so just like my dominoes are here set up, they're waiting. They're at resting potential too. But look what happens with the dominoes. If a stimulus comes along, and the stimulus is big enough to make that first domino fall, then they're all going to fall. But think about this domino. If I were to just bump it very, very gently, it would probably just shake and it would go right back to where it was. And if I bump it a little bit harder, it might shake a little harder, but it's going to go right back to where it was. But if I hit it just hard enough, instead of it shaking, it's going to fall over. So look, these are like those graded potentials that were too small. They were sub-threshold. But this one reached threshold. And if you reach threshold potential, it's going to fall over. The same thing happens here. Remember, our thing looks like this. That's resting potential. All of here, all the way across, it's resting potential. All the way across, everything's at resting potential. And then here comes my stimulus right here. Remember, it's going to be chemical or mechanical. And what it's going to do is it's going to make this go up. Well, if I don't reach threshold, it's just going to come back down, just like this thing happened here. And again, no matter how close I get to threshold, it's just going to come back down unless I actually reach threshold. But as soon as I reach threshold, I'm going to get an action potential. As soon as I reach threshold, this thing's going to fall over. And this action potential is going to create another action potential here. This domino is going to make that one fall, and then that one, and that one, and that one. They're all going to fall. And what's going to happen is, all the way down here, every domino is going to fall. And the same thing's going to happen here. I'm going to get an action potential over and over and over until I get an action potential that starts here, and that one creates that one, and that creates that, and then that one, and that one. It's going to go all the way to the end. And remember, they're all the same size. This one's plus 30. This very last one is also plus 30. Every one of them, plus 30. But think about this. If I were to set up dominoes, what do you think the longest distance I could set up dominoes would be and make them all fall down? Is it 1 foot? Is it 10 feet? Is it 100 feet? Is it a mile? Is it 5 miles? Is it 100 miles? How far do you think you could set up dominoes and make them fall down? Now, assuming you're on a nice flat surface, how far could you do it? Want to guess? It's unlimited. You could set up 1,000,000 dominoes and they would all fall down. And so it could travel all the way from here, from the US, all the way to China, assuming you could cross the ocean and all that. If it were this nice flat surface and they were all set up correctly, I could just hit one domino, and some guy over here in China could wait and wait and wait, but eventually that domino would fall, and then that would be a signal to that person. The same thing happens with action potentials. The distance they can travel is unlimited, just like those dominoes. And so this signal, which starts here, even though it's the very last one, this is going to be this signal at the very end of this membrane. And so neurons can be long. Think about a blue whale, which is the largest living animal. Blue whale, from their brain to the tip of their tail, how far do you think that is? I don't know, 150 feet or more. And yet an action potential that starts here can travel all the way to their tail to tell their tail to move. I wish you could see these things, but if you do the PowerPoints, you will. But if I step on this balloon right here, what's going to happen is the air is going to go this way, and it's going to bulge. And that bulge is actually going to travel down this balloon, just like this, all the way to the end. It's going to travel like a wave. And the same thing's going to happen here. If I get an action potential, it's going to travel all the way to the end, sort of like a wave. And remember, it's going to travel all the way to the end, be the same size. The action potential can travel long distances without losing any height, without losing any information. Any questions about action potentials? So when we look at that, there's this set of like dominoes that are set up. And if I bump one domino, they're all going to fall down. The same thing's true for this action potential. If I create one here, then I'm going to create a bunch. And every little part of this membrane is going to go through an action potential. It's called continuous propagation. So continuous propagation is this set of very small steps that happen over and over and over and over as the action potential spreads. It's kind of like if you took a person and you told them they had to walk across the floor, and you've got to touch every bit of the floor as you go. So the only way to do that is to put one foot right in front of the other, and then move that foot around right in front of the other, and then move the other foot right in front of the other, like this. It's a bunch of very small steps to get all the way across this room. All the way across. You're just going to have to keep doing that over and over. In other words, it's lots of little steps, and it's slow because it's lots of little steps. So if you look at it, it's like these dominoes, like we've said before. Every domino is going to fall. It's a bunch of individual steps. Here's one step, 2 steps, 3 steps, 4, 5, all the way. Lots of little steps. It starts at the very first part of the axon, which is right here, and then it spreads. And you can watch the action potentials happen with electrical activity. You can watch it. And remember, it's because the ions are coming in and the ions are going out. And it just spreads down the whole length of the axon. And so if you look here, you can see these channels are open right here. And this one's going to cause an action potential to form here. So the action potential is going to move a little bit. And this one's going to cause an action potential to form here. And so it's going to move. Don't forget there's a reset period. That's the refractory period. And so the action potential, even though sodium moves this way, I cannot get another action potential. I cannot right here because it's in the reset period. But this one over here is already set up. And so the action potential moves this way. And then here, this has to be reset. So the action potential has to move this way. But look, by the time I get over here, this has already been reset. It's been picked back up. The domino's standing straight up again. It has been reset. But this is in the reset period. This one has never done, and so the action potential goes this way. But look, I can have another action potential here. So you can have one action potential following another action potential, following another action potential, following another action potential. I can have them going one right after the other, but they only go in this direction. And that's because of this reset period. It's because of the refractory period. So I get one-way flow. and you can watch it move. It's like a wave going down the membrane, just like dominoes. So again, if this domino falls, then this one's going to fall. But this one is going to be like this now. I can't make it fall again until I reset it. But once I reset it, I can have another action potential or another set of dominoes falling right behind it. So you can have one action potential, and then right behind it another one, and right behind that another one, and right behind that another one. And so you can have lots and lots of action potentials, but they only go one direction. And again, that's because of this refractory period, this reset period. Any questions? So, if we step on this, we're going to get this action potential. This going to bubble thing is going to move the air. Same thing here. We're going to get an action potential, and then the action potential. And remember, every little part of the membrane is going to go through an action potential. And again, the name for this is continuous propagation. Well, this happens when we don't have myelin. So we get continuous propagation. It happens on unmyelinated neurons. But some neurons have myelin. Remember myelin? We have this myelin sheath, and there's two different types of cells that can make myelin. The oligodendrocytes that you have in the central nervous system and the Schwann cells that you have in the peripheral nervous system. Well, when you get those, look, it doesn't look like this. That's a naked neuron. It looks like this, and it has myelin around it. Remember, myelin is insulated. But these cells, remember, don't touch each other. We have these nodes of Ranvier, these nodes. And so what's going to happen when you have an action potential here, this one, every little part of the membrane has to go through an action potential, every little part. And so it takes a long time. It's slow. But what happens here is the action potential looks like it's jumping because only the nodes go through action potentials. It looks like it's skipping. And the word for skipping is saltatory. So here we get continuous propagation, but here we get saltatory propagation. And it's much faster. So here's saltatory propagation. The action potential happens here, and then it jumps, and then it happens here, and then it jumps. So remember telling you about my guy walking across the floor. When we did continuous propagation, he had to put his foot so that their feet touched every bit of the floor. So it was one little step, next little step, next little step, next little step. And it's very, very slow. But if it's saltatory, the guy can take big steps. So he can take one step and his foot touches here, and then take another big step and it touches here, and another big step and it touches here. And look, it takes fewer steps, and so it's faster. And so when you look at myelinated neuron, it's almost like taking giant steps across the room. Not tiny little steps, but giant steps. And the reason it happens is because there's only channels in the nodes. Look, this part, here's the myelin. It's insulated, and there are no channels there. And so the action potential only happens where the channels happen. And so instead of taking, I don't know, like 8 action potentials to get from here to here, like this. If it were a naked neuron, it would take that many action potentials. It only took 3. So saltatory conduction is much, much faster. And remember, it happens only at the nodes of ring VA because that's where the channels are. So it looks like it's jumping from node to node to node. So if you compare the other one to this one, look, here's some sort of bind tape or something bound around this balloon. And so if I mash on it here, that can't get up. And so what's going to happen is the bulges are only going to happen here. Same thing here. When I get an action potential, it can't happen in here. It's closed. There are no channels. So the action potential only happens here. Again, this is another little video that you should watch. But action potentials on myelin sheaths are much faster. They can be as much as 300 times faster. So if you compare unmyelinated to myelinated, look, they started exactly the same time. But after 10 milliseconds, this has only made it this far on an unmyelinated neuron. There's a bunch of action potentials, like tiny little baby steps. But after 10 milliseconds, look, it's made it this far on the myelinated one because we only have this many action potentials. So much, much faster. So if you're watching the PowerPoint, you can see the comparison of these two. So this is going to be, again, every little part of this balloon is going to bulge. But here, it's only going to happen here, 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 and here. So I don't know how many action potentials that is, but let's say it's 30. But here it's 1, 2, 3, 4, only 5. So look, in the same amount of time, 5 went this far instead of it taking 30 to get this far. So back to the characteristics of action potentials. Remember, they're all or none. You either get one or you don't get one. In order to get one, you have to reach threshold potential. Once you do, though, they're all the same size. They don't dissipate. And their size has nothing to do with the stimulus as long as you reach threshold. And then remember, they're recreated just like dominoes falling over and over and over down the membrane. And then don't forget, they have a refractory period, this reset period. And that makes sure that they only flow in one direction. So here's another little summary slide. And what this does is it allows you to compare graded potentials with action potentials. So let's go down this list real quick. If you look at graded potentials, remember it was chemical gated and mechanical gated. If you look at action potentials, it's just voltage gated channels. If you look at graded, remember it's mechanical and chemical channels, and those are only found on dendrites and cell bodies. With action potentials, it's voltage gated channels, and those are only on axons. And then, remember, these can go towards 0 or further away from 0. But action potentials only go towards 0. Now they come back down, but you must have a depolarization before you can have a repolarization. This one, they can start off and then go in either direction at the beginning. And then remember, graded potentials vary in size, and the size is dependent on the stimulus strength. Action potentials are all the same size, and their size is independent of stimulus strength as long as you reach threshold. Then graded potentials, there's no refractory period. It can happen over and over and over again, one right after the other. But action potentials have a refractory period. And that's what produces this one-way flow, one-way transmission. And then graded potentials only travel a very short distance. So they're only good for short distance communication because they dissipate. But action potentials do not dissipate. And so they're good for long distance communication. And then one other thing, which we will see a little bit later on, graded potentials can add together. Action potentials cannot add together. They follow one after the other, but they cannot add together. Here's that graph again. Again, you should practice. What happens at 1? What happens at 2? All the way through until you can just put them in order and talk about them. And then the last thing, remember we said that you have this neuron, this axon, and if it's naked, remember it's slow. If it's myelinated, it's faster. But the more myelin you have, the faster it is. And so different neurons have different velocities, they have different speeds, and so we classify them. And they're classified into 3 groups: type A, type B, and type C. A is the fastest, B is less fast, and then C is the slowest. Some A's can travel as much as 150 meters per second. That's like a football field and a half in 1 second. And some are so slow that they only travel about a half a meter per second. So look, this is 300 times faster. And then the last thing is, if all action potentials are the same size, which they are, if I'm the brain sitting over here or the spinal cord, and an action potential arrives, how do I know how big the stimulus was over here? Was it a big weight or a light weight? Was it a lot of chemicals or just a little chemicals? How do I know? Well, you can't tell action potentials apart because they're all same size. The only other way to do it is by how many action potentials there are. And so what your brain does is it sits over here and it counts action potentials. So look at this. Here's our stimulus, but it didn't reach threshold. That's threshold. If I don't reach threshold, look, I'm not going to get an action potential. And if my brain is sitting over here counting action potentials and no action potential arrives, I'm not going to know this happened. Not going to know. Have you ever looked down and seen a mosquito sitting on your arm and you didn't feel it? Well, the reason why is because he's not heavy enough. He didn't produce enough pressure to reach threshold. And remember, if you don't reach threshold, you do not get an action potential. So look, no action potentials. And if there's no action potentials, Nothing travels to the brain, and so this goes undetected, whatever it is. So think about yourself in a room that's dark. There can be enough or a tiny, tiny bit of light in there that you can't see it. So it's very, very dim. Some animals might be able to see it, but you can't. It goes undetected because for you, it did not reach threshold. But then look over here. Here's a stimulus that did reach threshold, but it's not a very big stimulus. But you are going to get action potentials. Look how far these action potentials go up. They go up to plus 30. Remember, they're all the same size. And what my brain is going to do is it's going to count action potentials. So look, only 4 action potentials arrived. And so my brain says 4 action potentials, that's a small stimulus. In other words, that's not a very big thing. It's not a very heavy thing. It's not a very bright light. It's not a very loud sound. But look again. Here, look how big the stimulus is. It's this big. And when I get a stimulus this big, I'm going to get this many action potentials. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 action potentials. And when my brain counts 11 action potentials, it's going to interpret that as a much bigger stimulus than 4 was. And so the sound is interpreted as louder. The light is brighter. The taste is stronger. The weight is heavier. It's a bigger stimulus. Over here, it's a very small one. It's a dim light. It's a soft sound. It's a weak taste. It's not very heavy. Our brain sits and counts action potentials. And this is called coding. It's called coding for stimulus strength. So it's not the size of the action potential, it's how many they arrive, the frequency of the action potentials. So that's how the brain determines stimulus strength. There are some things that can go wrong with neurons. One of the things that can go wrong is called multiple sclerosis, or MS. It's an autoimmune disease, which means that your own body attacks the neurons. And the part of the neuron it attacks is the myelin sheath. It attacks and kills oligodendrocytes. It does not affect Schwann cells. It affects oligodendrocytes. And if you destroy the oligodendrocyte in the central nervous system, the myelin sheath gets destroyed. And what it does is it slows everything down. And so we get all kinds of weird symptoms because the neurons are not talking, at least as quickly, to all the different organs in the body. The impulse gets slower, and as the disease progresses, it stops. And if you stop talking to those organs, then those organs don't do anything. And so as multiple sclerosis goes away, you get muscle weakness, loss of muscle control, you get speech problems, visual problems, you wind up with things like incontinence, things like that. There are some treatments, but they're not a cure. Okay, anybody have any questions? So that's where we're going to stop for today.